Okay, so today we want to talk about apoptosis, which is basically a programmed cell death. And apoptosis, we normally have two pathways. We have the extrinsic pathway and the intrinsic pathway. This extrinsic pathway is also called the death receptor pathway because it basically entails the use of death receptors. So remember that every cell usually has a surface molecule called a death receptor, which helps in the killing of that cell in case that cell is supposed to be killed, maybe due to an unfunctioning genome, okay? So we have two types. We have just two examples that I've highlighted here, but we have other examples. We have type 1 tumor necrosis factor receptor, and we have the first also called the CD95, okay? So this is a receptor. Remember the ligand will be called the CD95L ligand, L for ligand, also called the first L for ligand, first ligand, okay? So this receptor basically called the first usually contains three subunits, okay? So whenever this CD95 ligand binds to the receptor CD95, we normally have the trimerization of the three subunits of the receptor. So whenever they trimerize, the receptor will be activated and it will attract the intracellular adapter protein called FAD. Okay, then after it has attracted FAD, FAD will actually recruit procaspase 8. Remember, it's a procaspase. So we need to cleave it to form a caspase, which is the active form. Okay, so this procaspase is usually cleaved to form smaller subunits which generate the caspase 8. Now, this caspase 8 is very important because it will activate downstream caspases, such as caspase 3, which is usually very important in cleaving the DNA and other substances to cause a cell death. So remember, it is caspase 3 that will destroy or cleave the DNA, okay? And whenever it cleaves the DNA, will actually lose the cell will die, okay? I want us also to understand that we also have another important protein called the FLIP, and FLIP basically prevents the actions of all these caspases. So whenever FLIP has been expressed in a cell, that cell won't die by apoptosis, especially by the extrinsic pathway, because the caspases want to be activated, okay? So another important thing to understand about this pathway, the death receptor pathway, is that caspase 8 might activate the proapoptotic member of the BCL2 family called BID. We'll understand so much about the BCL2 family when you're tackling the intrinsic pathway in just a few minutes to come, okay? So whenever we activate the BCL2 family, remember we are going to the mitochondrial pathway, also called the intrinsic pathway, right? And this intrinsic pathway basically involves things like cytochrome C, which we'll talk about in a few minutes, okay? So just know that this caspase 8, number one, can bring about apoptosis by activating downstream caspases like caspase 3, which will cleave the DNA and other substances to cause cell death. But alternatively, it can kill the cell via activating the mitochondrial pathway. So it's a dual thing, okay? So we have some viruses which usually produces FLIP, homologs of FLIP. Remember, we said that FLIP will prevent apoptosis by preventing the downstream caspases from enacting their action. So we have some viruses which usually produces homologs of FLIP, okay? So when they produce these homologs of FLIP, it means that they can't be killed. The infected cells can't be killed by apoptosis because this FLIP will prevent the action of the downstream caspases that I just talked about, okay? So let us go through this diagram quickly. It is quite simple to understand. We have the CD95L ligand, or the first L, which will bind to the receptor, which is the first. Remember, the first is made up of three subunits. You can see it from this diagram. Then whenever this ligand binds to the receptor, we'll have trimerization of this receptor. They'll come together to form this trimer, which will now be activated. It will be an active receptor. So this receptor will basically attract the FAD, and this FAD will attract the caspase 8, okay? So when it attracts the caspase 8, actually it will attract the procaspase 8, which will be cleaved to form a caspase 8. But this diagram is just trying to simplify the entire thing, okay? So we have said that this caspase 8 can act, look at the pathway. Let me use a separate thing. This caspase 8 can act by activating this caspase 3, okay? So by activating this caspase 3 can act via the card of the ICAD to bring about DNA fragmentation, okay? In other terms, this caspase 3 will degrade the DNA and other substances in the cell, leading to the cell death, okay? But again, we said that this particular caspase 8 can also act via the intrinsic pathway by activating an important member of the BCL2 family called BID, okay? So when we activate BID, again, will lead to another pathway that will lead to apoptosis. But I don't want to tackle this pathway because it is what we are going to talk about in a few minutes, okay? So for this diagram, I just want you to understand that that when this ligand binds to the receptor, it will be activated by trimerization. FAD will be attracted, which will now recruit caspase 8. Caspase 8 will go and activate caspase 3, which will go and break the DNA, and this will lead to the cell death. That is basically the death receptor pathway. So let us go to the intrinsic pathway, also called the mitochondrial pathway. Okay, so this pathway is usually triggered by a variety of stimuli. Okay, for example, we can talk about stress and even injury of the cell. Okay, and this pathway basically entails it basically entails making the membrane to be permeable. Okay, the mitochondrial membrane to be permeable. Okay, and this one will enable for the extrusion of cytochrome C. And I think we'll understand more about this mechanism in just a few minutes. Okay. So the integrity of a mitochondrial outer membrane is usually regulated by a pro-apoptotic protein. So I want you to understand that for a cell, we normally have the pro-apoptotic bodies and we have the anti-apoptotic bodies. So the pro-apoptotic bodies will basically facilitate apoptosis, while the anti, from the name anti, it will prevent apoptosis from taking place. Okay. So we have two types of pro-apoptotic proteins. We have the backs and we have the back. And we have for this particular slide, I've just put two examples of anti-apoptotic proteins, but there are others. You can just go and look at them. But we also have the BCL2 and the BCLXL, which are the most important member of the anti-apoptotic protein family. Okay. So when you read different books, they say that the pro-apoptotic bodies and the anti-apoptotic bodies usually exist in a balance. Okay. So it means that when you degrade the anti-apoptotic body, the pro-apoptotic body will be upregulated and it will actually enact its action. On the other hand, when you break down the pro-apoptotic bodies, it means that the anti-apoptotic bodies will be unopposed. Okay, so they'll actually enact their functioning. Okay, so basically, for us to have apoptosis, we need to have the pro-apoptotic bodies, but not the anti-apoptotic bodies. Because remember, anti-apoptotic bodies are basically preventing apoptosis, but we aren't apoptosis. So we need to degrade anti-apoptotic bodies so that we can remain with pro-apoptotic bodies. Okay. So the pro-apoptotic proteins promote mitochondrial permeabilization, and the action is inhibited by anti-apoptotic proteins. Pardon me, this word is giving me a lot of headache to pronounce it, but it's okay. You're getting the concept anyway. So these pro-apoptotic bodies are the ones that are very important to create pores in the outer mitochondrial membrane so that cytochrome C can leak out. Cytochrome C can leak out. Okay. So let us continue. Okay. So we have a very important family of proteins called the BH3 only proteins. Remember the BH3 only proteins. And we have three examples. We have the BAD, BID, and the PUMA. So what these three proteins do is quite simple. They break down the anti-apoptotic bodies. So these three will come and break down the BCL2 and the BCLXL. So when they downregulate BCL2 and BCLXL, it means that the functioning of the pro-apoptotic bodies will go unopposed. So we'll have apoptosis. I hope you're getting it. So basically, once you have degraded BCL2 and BCLXL, which were anti-apoptotic, so it means that these backs and back will be activated, okay? And when they're activated, they lead to formation of pores in the mitochondrial membrane, and this one will lead to leakage of the cytochrome C, okay? So when this cytochrome C has leaked out of the cell, it will go and bind to a very important protein called APAF, okay? So this APAF, APAF, will go and activate caspase 9, okay? And this caspase 9 will just do the same functions as caspase 8 in that it will go and it can even activate caspase 3, and caspase 3 can now lead to degradation of the DNA, which can lead to apoptosis. So basically, it is the same thing. It is basically a repetition. So maybe just to give you a quick recap, we have the components of the intrinsic pathway. Number one, we have caspase 9, we have caspase 3, or we have the caspases, we have APAF, APAF, we have cytochrome C, we have the anti-apoptotic proteins. Examples are BCL2 and BCLXL. We have the pro-apoptotic bodies, which are the backs and the back, okay? And you have the BH3 only proteins, which are the BAD, BID, and the PUMA. And remember that we also have a very important organelle in this pathway called the mitochondria. Those are the components of the intrinsic pathway. And I think I've talked about how they bring about apoptosis. So maybe you can just go through a diagram quickly so that you can understand better how this one works. So we have a simplified diagram here trying to show us how this pathway goes. So we have maybe a DNA damage or we have an apoptosis signal, okay? Now this one will actually be like an activator to the bud and the puma and the bead and the noxa proteins, basically the BH3 proteins, okay? And I said that the BH3 proteins usually cleave the BCL2 and the BCLX, okay? So when they cleave the BCL2 and the BCLXL, we'll have the backs and the back protein being unopposed, so they'll be activated, okay? So once the two of them are activated, they'll come and act in the mitochondria, and then will create pores in the outer membrane of the mitochondria. So cytochrome C will leak out, okay? So when cytochrome C has leaked out, it will go and bind to APAF1, okay? When it binds to APAF1, APAF1 will recruit procaspase-9, which will be broken down by cleavage to form caspase-9. Then this caspase-9 can now act downstream by activating other procaspases like procaspase 3, 6, and 7, which can now go and break down the DNA. And after breaking down the DNA, the cell cannot survive without a DNA, so the cell will die. Okay. So those are the two pathways of apoptosis. Yeah, I think I'm done and it's quite clear.